Hello. So we'll start the program. The more people are coming and joining. Of course, we started with the coffee outside and some more drinking. Okay. So they'll join. I have told them that we are starting the program. In your first part is only a few minutes of introduction. So today is Darwin Day, and it is important. Well, actually 12th February is the Darwin Day. We could not hold it on around that place, around that time, and this was the best time it was made available to us. And on behalf of Tamil Nadu Science Forum, PSL group, Popular Science Lectures group, I invite all of you. And those who are attending here and also through Zoom and other ways. So I invite all of you. The Darwin Day has become sort of interesting in Tamil Nadu. That is one of the things. Suddenly one of the VIP from Tamil Nadu started making remarks about the Darwin's proposals itself and how it affected badly Indian culture, Indian this thing, and there is some anger among scientific community that remarks are made by who are called VIPs. I will not name them, you probably know them, and it's unfortunate. We don't worry so much about it, and we will move on. Of course, Darwin made extraordinary contribution, and the evolution has played such a role in our growth, in our life. We are all there because of that, and that is the reason we are talking about Darwin. Now, today we have two people speaking about to us. Dr. Geeta is actually known to us earlier. She is not new. And she is also one of the founding members, various people who were involved in the starting of the Tamil Nadu Science Forum itself. Around 1980s, when research scholars in Chennai, many people and others, we were inspired by Kerala Sastra Sahitya Parishad and wanted to have this kind of program in Chennai. So it turned out to be initially a local Chennai program. We tried to go out to some other places, but mid-80s and later 90s, it took a lot of new directions, like literacy program, health program, women's programs, etc., which somehow got linked with the issues about science popularization, which at primary school, middle school, and other levels. So the Science Forum has grown, and we are very happy. There are a lot of volunteers now all over Tamil Nadu. I understand of the order of some 25,000 members are there. And there are journals, magazines for primary level. It's called Tulir. And middle school level, we have also magazine. So in English. So there are various things of this kind we have been doing, but this kind of program, Popular Science Lectures, is more or less specific to Madras or Chennai, where earlier in 2018 we started doing, and several places like Anna Centenary Library and IIT, Math Science, IMSC, we have held several meetings, and this is 55th meeting, right? 58th meeting, and we are very happy. We have been consistently holding it. Of course, during COVID, it turned out that we had to hold it online, which we were successfully conducting it. Now it is interestingly that lesson has given us that it is online and offline, hybrid mode. So the interesting thing about it is it was not limited to Chennai people who come and attend here. So now the participation is from all over Tamil Nadu, many places, including few people from North India, Delhi or Kanpur or something, people who know about it, they do attend, take part in the program. So this is as far as Tamil Nadu Science Forum is concerned. And Dr. Geeta was known to me at least for 40 years. And we were involved in the Tamil Nadu Science Forum right from the beginning. And we did various activities. But she then moved to US. She was in initially in Arizona, Tucson, later to Stony Brook. And then after that she moved back to Delhi University. She was in Delhi University as a professor for a long time, for substantial time, and then retired from there. And now she is back in Tamil Nadu to carry forward Science Forum activity, which she started, one of the people who started it. And I'm very happy to have her again in our program. Now I will, the first speaker is going to be Geeta. The second speaker will appear on the screen. He's from Canada, and you will probably introduce him to the audience. Okay? Thank you. Thank you. Now let's have Geeta.
Good inauguration. Is that okay? All right. Thank you for that little introduction, and thank you for inviting me to do this. Before I get into what I want to talk about, I just wanted to introduce you to the other kinds of activities that I have been involved in at the University of Delhi, and then just this year in Chennai. And my intent in putting this up is to those of you who are not at the celebration on the beach this year. Please be inspired to do this again next year, and I hope that other people across Tamil Nadu will also think about it. We'd be happy to help you in whatever way we can, remotely or otherwise, to enable this to happen, because my experience with students in Delhi is that it was a really good way not only to popularize science, but for the students to sort of get involved in such activities and actually participate in talking about it. So I strongly urge this activity also. So I just wanted to very briefly, in a sense, this is Evolution 101. So I just want to briefly talk about what Darwin was about, what Mendel, the geneticist, was about, and how it came about that scientists were able to put together these two lines of discussion and research to finally, well, I won't say finally, but to certainly come to a more comprehensive understanding of what evolution is about or could be about. So everyone is familiar with Darwin, I think. Most people may not be familiar with Wallace. In fact, the two of them, I would say, simultaneously pretty much presented to the world this theory of natural selection. And indeed, Darwin had spent so much time preparing his work, The Origin of Species, that in the meantime, Wallace sent him an abstract from Indonesia where he was, and told him, look, this is what I think, what do you think? And Darwin said, this is exactly what I think, so what to do? He got very agitated. And so simultaneously at the Society for the Linnean Society, these two papers were presented. One is Darwin's paper and the other was Wallace's paper, both pretty much saying the same thing. Now, how did they come up with this idea of natural selection? For one thing, everybody knows the diversity out there. Well, not all of the diversity, but that biological life is extremely diverse. There are many different kinds of plants and animals. Equally, within a species, and this is a kind of an exceptionally variable species, sometimes within our own species, there will be a lot of variation, right? Now, putting all of these things together, this observation on variation out between species and within species, and putting it together with Malthus, who was a population demographer, I guess, who studied populations in the, actually it turns out his first publication was in the end of the 18th century, 1790 something, and he repeatedly came up with different versions of his hypothesis, and this is the 1826 version, which I presume would be the one that Darwin must have read. But what did it say? Basically that populations grow, this is about human populations, that populations grow, that resources are limited, and that's where a lot of our some of the problems might come from. I'm not going to be talking about what Malthus said, but what was taken away from nature and Malthus by these two scientists was that firstly there is a lot of competition for resources out there, right? Because there's many more organisms than there is resources available to them, so there is a high degree of mortality in populations of organisms. Now, individuals who have good traits, better traits that allow them to survive better and reproduce better will do so, right? Now, because they reproduce better, this trait is going to spread through the population over generation. The next generation it will increase, and over many generations, and therefore over a long period of time, it may come about that this trait completely dominates the population. It is present in high frequency in the population. So this trait, such a trait is called an adaptation. It is the trait that allows that species, individuals of that species, to exist and multiply better. And we have said that we will say that the population has evolved, right? So the individual doesn't evolve, it's the population that evolves. It's a population level phenomenon. Now, it has been called by Ernst Mayr as one long argument, okay? So how do we say that high mortality occurs? Because we know organisms can multiply enormously. We also know that organisms, population sizes remain stable. If elephants were to multiply at the rate that they might, they would simply very soon completely cover the planet. So that obviously does not happen. And because natural resources are limited, there is competition and there is high mortality. And because there is competition and because individuals may differ in what their properties are, the ones which have better properties that allow them to survive better will survive and reproduce. Now, so that is what we will call natural selection. Okay. Now, if the traits have a genetic basis to them, then this trait will carry over from one generation to another because the genes are being passed on from parents to offspring. And after many generations, this population, because it shows this change, we have said that the population evolves. So the genetic component is very important. Now, the problem was that Darwin did not really understand, or generally we did not understand genetics at that time. Remember that this was 1859, and yet at that time Mendel had already come up with his ideas, well, not a little later, he had published in 1866 about his ideas of genetics. Now, at that time, how we had sort of vague ideas about what inheritance is. So in Tibet, adults, people who live there, can breathe normally, even though the altitude is so high. If you and I went there, we shouldn't firstly go there just like that. We should go slowly, acclimatize ourselves at different altitudes, so that finally when we reach a very high altitude, we can actually function properly. Now, the Tibetan adults can actually pass on this property of being able to function normally to their children. They do, we know that. The question is, what about visitors like us who go there? Can we, once having gotten the ability to breathe like well in at high latitude, altitude, can we actually pass on the property to others, to our children. Now, at that time it was thought that yes, it was possible. That whatever you, for instance, there are many traits that we might acquire during our lifetime, and people thought that in fact we could pass it on to our children. And it was not clear, therefore, as to what actually was happening. Now, so Mendel was the one who came in and sort of showed that in fact if you have plants which may have white flowers or purple flowers, and you cross them and they produce, if they are lines that produce only white flowers or purple flowers, and if you cross it, what you might get is a purple flowered plant, which he showed in peas. Now peas, if you cross those two peas, this is basic genetics that many of you might have had. If you get to cross those two peas, then you'll get back the white and purple flowers in a ratio of 1/4 to 3/4, 25%. Now, by doing counting the numbers, etc., Mendel postulated that there were these so-called factors which were underlying the inheritance of this trait, that the one trait was hidden, its effects was hidden in the hybrid population, which we know that will have the trait of, will be carrying both the traits. But that is hidden and it shows up in the next generation. So these were called, the appearance of an organism is called a phenotype. The factors, he called them factors, the name genotype, genes and alleles and genotype came up later in the 20th century, early 20th century. And it was shown that many such traits actually are inherited in this fashion. There's many complications that we will not go into. But on the other hand, there are many traits which show continuous variation. So you can't say, this is in these seeds of the wheat grain. There are many variations in color. Okay, it's very hard to distinguish them. How many genes are there? What's happening? What is going on? So at that time, it was not clear that these kinds of genes were also important for these kinds of continuous traits. So there was a big battle towards the end of the 19th century, early 20th century, between the people who were studying quantitative traits or continuous traits and those who were studying the kinds of Mendelian traits which you can distinguish qualitatively. As to what the mechanism of inheritance is. Then there was a Swedish scientist, geneticist called Nilsson Ehle, who studied these wheat germs lines and showed that in fact there are multiple genes which all contribute to color. So if a lineage, can I use this? Okay, if one variety has this, can you see that? Okay, this one has 6 copies of the one type of allele which makes the color. This one has no copies of the allele or the form of the gene that makes the color. Okay, so you either have white or red. Now if that were the only thing, that would be okay, but because you have different combinations, you have this whole gradation of colors, and that's what you'll see in this fashion, that it will show up like a normal distribution because they're just the different traits are varying independently. But the question comes about as to why, when you plot this distribution, you won't see distinct categories. The best fit will be a normal distribution, right, a curve. So where does such a curve come about? Why is there a blurring between one phenotype and the other? And the insight that was offered was that in such different phenotypes, there is a certain smoothing because of effect of environmental. So every phenotype, every genotype will not develop in exactly the same way in all the time in a particular environment. There will be some small environmental variables which will actually affect what comes out. So it's a bit of a, not exactly stochastic, but definitely there's a, a statistical pattern to what's going on. So this kind of smoothing effect of environmental variation raises the question, if you see a distribution like this one, right, how much of the variation is due to genes and how much is due to the environment? And that was the big question. So this advance was actually made possible by the studies of scientist called Fisher. We'll just come to that. But then what he was able to show us was that the phenotype, the variation in the phenotype, or the phenotypic, what we see, is a consequence of both genetic and environmental factors. And the point of interest is to understand how much of this is due to environment. So we understand, therefore, through this work and other work, that we can actually understand this variation in phenotype in terms of how much is genetic and how much is environmental. Not the variation itself, how much is due to genetic and how much is due to environmental factors. So Fisher was the one who then showed, and this is probably a method that is many people are familiar with. He was the one who introduced the idea of the analysis of variance. So he invented the term and the concept of variance, showed that the variance is a good measure of population variability, and that the variance due to in the phenotype is composed of the variance due to the genotype and due to the environmental factors. And the task for a geneticist or a breeder, because that's where he started working, he did this work as a school teacher, and then he was, I suppose, inducted into the, I think in Edinburgh, in the breeding institute, animal breeding institute, where he continued his work. And so the question is, when you are trying to breed, you see variation. How much of it is due to genes and how much is due to environment? We want to pick those traits which in fact are due to genes, not the ones which are due to environment. So this partitioning of variance is something that's used in many situations, but here's where it kind of originated. So if you partition this variation and you look at just at the genotypic effect, like the variance due to genotype. So there's going to be variation due to the genotype, variance due to the environment. The genotype variance is actually made up of multiple components. So one is called the typical dominance effect that we are familiar with, right? Where one allele hides the effect of the other. So what happens when when you're crossing two parents, that the one that you want, the purple color, or say the white color, if you want the white color, it disappears in the second generation. Then how are you helped? It's very difficult to then carry on your breeding program if that's what it is, right? Now I'm talking about artificial selection, people wanting to select from something for some trait. So there's the dominance effect. Then there's also effect. So that is, effect is due to two alleles within a gene interacting within the activities of a gene. Now there may be different genes that actually interact with each other. One may hide the other, it may affect the other. So all of that again is a kind of effect that will disappear in the next generation. So, but there are genes, additive genes like the ones we saw in the previous slide, where each gene is actually contributing and increasing the effect of the, cumulatively they have some effect. So this kind of additive effect is the one that we want, because that is the one that will actually appear in the next generation. So if to make a cross, see the trait, whether it's there in the, it will be there in the next generation. You have to see how much of it is there, is due to additive effect and how much is due to other effects. So the proportion of the variance that is due to additive effects and that is due to environmental, of the proportion of the phenotypic variance that's due to additive effects is the one that you're interested in, because that is the one that reflects the trait, the additive gene, which will pass on the traits to the next generation. So that's all very well. So this is called heritability. So we want higher heritability, or we want to take traits which have high heritability. But how do you find out? How do you estimate it? That was a big question. I mean, as we know about the concept of the mean in populations, estimating the mean is another big question, right? So here there are multiple ways that have been suggested. One is that it can be estimated as the slope of the regression between parents and offspring. So if parents have a certain trait and offspring have show the same variance of the same trait, then you'll expect that the variance should be high if heritability is high and low if heritability is low. So you may have different kinds of patterns. So here there is heritability is 0 because there's no correlation, between the offspring trait and the parental trait. Here there is heritability, and here heritability is very high, which means that in that population there are many additive genes for that trait. We are not talking about how many genes. So here you see, in order to do this kind of analysis, we don't need to know how many genes, what are each of them doing. We are just partitioning the variants into this and that and getting a sense of what the heritability is. Now, so studies like this have been done in the wild, where doing some kind of breeding, manipulating the breeding, people have been able to establish that in these song sparrows in the wild, the heritability is very high. It is 0.998 or so almost 1. So that means there's a great deal of heritability in the trait measured is beak depth. So how large can the beak get? So there's a great deal of variation. So if there is variation in beaks, then there will be a lot of change, because there is, the selection can act on that variance. Now, another way to estimate, with the same thing can be done with artificial populations, which you can breed in the lab and do the same thing and estimate the heritability. Now, this kind of artificial selection can actually lead to an enormous gain, increase in a trait of interest. So in this case, this is a report of an experiment started in 1889, which was, periodically they've been publishing the results. This is after 100 generations of selection. What happens to oil content? So from about 5% oil, after 100 generations, they've gone up to about almost 22-23% oil content in the. So that's an answer to why we have so much corn oil available in the world. There are varieties which have been bred to actually yield a lot of oil. Now, and equally, they also selected to get less and less oil content, and that can't go below 0, and that within 85th generation they had reached that. But they also did an interesting thing, which is not so important, but just an interesting thing, that midway they started selecting again, and find that in fact you can actually select again for high yield, even though it's gone low. You can take it from one of the low content oil and again heal. That means the variability has not disappeared, or it has appeared again. We don't know what the factors are. But the point here is that comparing these heritabilities in selection experiments, you can actually come up with two quantities which are called comparing the response to selection in these selection experiments, you can estimate heritability by looking at how much this so-called selection differential was. That is, your aim is to go from the parental mean population of from here, in this case it is I think 5, to you want to go to 7 yield, okay, whatever it is. But so that would be the selection differential, okay. Now in the one case you might get no response at all, right? In which case the response to selection is 0. In another case you may get a response of just 1. So although you're aiming for 2, and how do we aim for 2? You're selecting those parents who have 2. The quality is 2% or whatever it is, and you're breeding them, and you find that the progeny don't have 2 but they have 1. So that's the response to selection. So that's another way in which they measure heritability. That is, as a ratio of the response to selection to what the selective pressure or the selective differential is between the parental population and the breeding population, the one that's selected, the selected population. So in the first case, there is no heritability, which means that additive variance is 0. So the additive genes are probably very low. So this is a mistake. I shouldn't say VA is absent, it's just 0. Whereas in the other case, additive variance is there and is quite substantial. So that's why there is response to selection. So in this case of the corn, the maize study, they actually measured these heritabilities across by looking at their data, right? How much selection pressure was applied, how much response to selection was there, and they found that there was a, as the oil content, or rather as the number of generations is increasing, the heritability is actually decreasing, which means that selection is acting on the variation that is there and effectively is used up all the variation. So unless you have new sources of variation, you won't be able to make any more improvements. However, and that's also the case when you are trying to go towards low levels of selection. Low levels of oil. Okay, so that's the other way in which you can count heritability. So, estimate heritability. So, through these quantitative genetic studies and experiments, it's possible to go out into the field, study the phenotype, and look at parental and offspring traits and get an understanding of what the heritabilities are, and therefore whether you can expect evolution to happen or not. Nowadays there's a great interest in people, Dr. Henry will be, I'm sure, talking about his work, but there's a lot of interest in what the effect is of human activity on this kind of heritability, variation and selection. So just to summarize this, what we've talked of so far, basically natural selection and also artificial selection. By the way, what Darwin had spent all his time doing when he was not publishing his work was doing experiments, artificial selection experiments, to substantiate what he thought he saw in the field, what the explanation that he thought existed. So some traits increase survival and reproduction, which we call fitness. So if there is variation in this trait, in such a trait, then the better variant will be selected because of the effect of the selective forces. And that is what we call natural selection. Now, if that better variant has a genetic basis, then it will be passed on to the next generation, and over time that property, that trait will increase in the population, and that is what we call an adaptation. So nowadays in evolutionary biology, people try to not talk about adaptation as a process. The process of natural selection with genetic basis will lead to adaptation. It's just less confusing to think of adaptation as a noun, right? It is an adaptation. What trait is the adaptation? That's how we want to think about it, but that doesn't always work. So this is what Mendel and Wallace had come up with, right? And then of course there was Mendelian and quantitative traits. Like I said, there was some discussion about what the whether all kinds of traits have a genetic basis of the kind that Mendel described, and the answer is yes. There's no blending inheritance, because people said, they said if you're looking at these quantitative traits and you keep on, then it's just going to blend one into the other, because it's never going to, because inheritance is blending. How do you expect it to change in any way, because it's just going to keep reaching the midpoint? And the answer came from this the quantitative genetics work. So it's also it is a case that the evolution of both types of traits have to be investigated, or are being investigated through statistical means. Now it's all become very, very sophisticated, and that is statistical methods are used to study this. So I'd just like to leave you with this, that selection can have a really dramatic effect. Now if you if you saw a cabbage and a cauliflower and a kale and broccoli and so on, I don't think unless of course if you taste it, you might say, oh, that tastes like this, but it's the same species, exactly the same species. So that is something clearly people find fascinating, especially these kids at the Delhi University Flower Show. And I think it's not as dramatic as you might think dogs are, because you still recognize dogs, right? You may not recognize that a cabbage is the same as a cauliflower. Anyway, I'll leave you with that idea, and thank you very much to IMSC and TNSF for organizing this, and I will hand you over to Dr. Professor Henry. You want me to give an intro? Okay.
Thank you. Hello. Thank you, Geeta. One small instruction. Okay, sure. So some of you may have questions. So if any of you want to ask question, can ask 1 or 2 questions. We will allow more questions after the second speaker, Adri, finishes. And about the Science Forum, you want to say something?
I want to see the questions in the network.
Okay.
No question.
Okay. We can take questions on YouTube, Zoom, or anything in the chat box. Okay, in the chat box, you can send your questions, and it will be reported to the speakers also. And if you have any question, you can ask me. Yes, go ahead. Go ahead.
Hello, nice talk. So I have a question about this heritability factor you have talked about, H square thing. Is nature try to make it like towards 0 or 1? I mean, nature want variation in the next generation, or it want the same as something like parents has, or?
You want. If you're selecting for a trait, you want it to be heritable, right? You don't want it to just disappear due to gene interactions. Therefore, you want something that will show up in the next generation so that you can be on the right track, so to speak. However, I must qualify the statement that now with molecular methods, you're able to use molecular markers to track genotypes. So that is certainly something that's happening. So, but this talk is not about that. It was just about what the state of art at that time had developed to make the. Is that okay?
Yeah.
So Darwin and Mendel aware of each other's work?
No. Okay. Mendel sent his, this is the story, I haven't actually checked up on that. So that's why I was careful to say that the disciplines met but not the scientists, right? So it was through the meeting of the disciplines and develop interactions that